Ladies and gentlemen, please do take your seat for our next panel discussion. We're looking at epigenetics from environment to disease. And may I please welcome on stage, but perhaps after the tannoy, it does sound like we are in an airport. We're about to jet off, but no one can jet off just yet because we've got a couple more sessions. To conclude day 3 here at the Abu Dhabi Global Healthcare Week, on the main stage here at M42, we are addressing epigenetics from environment to disease. And to moderate this session, may I please welcome back on stage Albara Elkhani, the Senior VP Operations, M42. Thank you so much, Albara, for joining us again. Albara, how's the event been going for you so far?
First of all, apologize. I've been on this stage maybe 20 times today, but yes, probably fed up me. But no, it's been amazing. I think in this last session as well, how we've been able to incorporate very non-traditional elements into health and well-being of our community has been an eye-opener for me. So it's been very interesting.
Absolutely. And you opened today as well with a fascinating session in the morning and to continue. Now we have a good friend of our stage, Dr. Wael Elamin, joining us again, M42's Medical Director of Environmental Sciences and Wastewater. And Dr. Andrew Jeremijenko, who is, of course, the Capital Health Screening Center, Medical Director and Consultant Occupational Health. And Dr. Javier Quilez, who's Associate Director of Bioinformatics at M42. Gentlemen, please do sit under your seats and take it away. Thank you.
Thank you, Mandeep. I think this is an interesting topic because we're segueing whoever attended our environmental science earlier, and we've defined the sort of basis of our discussion as the environment has a causal effect on us as human beings. So now maybe by just taking it to a genomic level, stepping it down a couple of levels, from looking at more holistically now into more actionable elements that impact the environment. So maybe, Javier, if you don't mind, what is actually epigenetics from a genomic perspective? What does that mean? How do you see it from a bioinformatics perspective? Are there changes that are caused directly by the environment? Can you just give us a bit of a baseline in the simple terms of epigenetics and the definition of epigenetics?
Thank you, Albert. So I think by now we all know about genomics, which are the DNA, the 3 billion bases and the 20,000 genes or so. And these, it's fair to say that those will remain invariable throughout your life if I were to sequence you. Epigenetics are chemical changes on that DNA. And I think key to epigenetics are 1, plasticity, and 2, functionality. Plasticity in the sense that epigenetics will change from the moment you are conceived, because during embryo development, epigenetic markers will change so that cells will differentiate into brain, heart, or skin. And they will continue changing over your life. So actually, epigenetics is a marker of aging, but also due to exposure. Like if you start smoking, you have a diet rich in sugar, etc. And functionality, because those changes are not random. Actually, they are used to regulate, so that, for instance, if you were to be in an environment where food is scarce, epigenetic changes could be triggered so that they activate or cell machinery to extract more cell. So I think this combination of plasticity and functionality is what make epigenetics very relevant. And on the correlation with the environment, as I said, the environment where we are or what we eat can modulate our body response, and in some cases can cause or lead to disease.
So if we take the hypothesis that an environment can lead to changes in our genomics that could potentially lead to disease, is that also potentially reversible if we change habits and exposure to certain elements?
In principle, yes, and that's why one of the use cases or interesting things in terms of epigenetics is what's called the aging clocks. So there's like a rush now to identify what is called, or determine the biological age, right? So I look at your methylation and I can predict your biological age, and you can compare to your chronological age, and then you can use this as like a vanity exercise, but also you can use it to create awareness of something that you are doing wrongly, and therefore try to change that, and maybe check your epigenetics maybe 1 or 2 years down the road after you have tried to do some conductive measures and see if your biological age has become better. That would be like as a proxy. Whether methylation could be used that now I change your methylation and you, without going to exercise, you become younger or not, I think in principle is, but it requires more knowledge from our side.
Isn't there a pill now you can take that means you don't have to go to the gym, right? There is something that was recently announced. You can take this pill, you don't have to work out again because you're affecting your physiology. But thank you, Dr. Javier. So Dr. Andrew, as a consultant specializing in occupational health, can you give us some, again, examples specific, because what Dr. Javier was describing was the biology behind the changes in our genome based on environmental stimuli. But can we correlate the environment to these changes and potentially look at mapping this to diseases as well?
Yes, look, that's a good question. I like to talk about firefighters because they get exposed to a lot of carcinogens, a lot of dangerous chemicals, and those firefighters, you can actually see that it affects their epigenome. Basically, if you choose to be a firefighter, you're choosing to be a hero, which is great, but unfortunately you're also choosing to expose yourself to nasty chemicals that will affect your epigenome. And what Javier was saying was, you know, the DNA is the blueprint, but the epigenome, it can change. It can be a brain cell or a heart cell, but it can also be, it can be manipulated. And if you get those nasty chemicals, it can change your epigenome. I've got a quote here. It was, your fate is not written. Your fate is not written in a stone genome. You can choose your destiny through your epigenome. So what he said is, yes, chemicals can do bad things, but you can also choose a good occupation. For instance, we know divers who are exposed to hyperbaric pressures. They can sometimes live longer, and now we're using hyperbaric oxygen to actually make people live longer. So by choosing your occupation, you can choose a healthier epigenome or a less healthy epigenome. We know that firefighters are actually classified by IARC, the International Association for Carcinogens, as it's a carcinogenic, class 1 carcinogen to be a firefighter. Everybody should be reading these sort of lists. I know they're not that exciting, but it's got alcohol. Alcohol is a carcinogen. How many of us are exposing ourselves to carcinogen in a daily dose? Chimney sweeps, firefighters, tobacco smoke. You know, it's not what you do once, it's what you do every day. And so that's why occupations are so important. We can look at the healthy occupations and we can look at the unhealthy occupations, and through that epidemiology, we can actually tell you how to choose your destiny.
Very interesting. So it's not just about what we willingly do to ourselves, but also what we're exposed to within our working environment as well. And that's an interesting point. And obviously these elements are related to aggressive, maybe occupational environments, but I think there's a lot more, not benign is actually not the right word here, but there's other, what we consider less aggressive exposure to environmental factors that are definitely affecting our epigenetics as well. So thank you, Dr. Andrew. Dr. Wael, as a clinician and our medical director for environmental sciences, you are putting a foot in both worlds. You're sort of seeing how the environment is affecting us in the human environment of hospitals and care as well. So do you have any anecdotes that relate to environmental factors on humans and epigenetics?
So thank you very much, Albara. And I think one thing you learn in scientific methodology is always ask yourself, why? Why does this happen? And the best examples are always to look at our twins. They share the exact same DNA code. They basically the genome is exact. Yet, and they probably end up living in the same environment, in the same house with the same parents most of the times, exposed to the same foods. Yet you may find one of them able to run a marathon at 50 and the other one is bedridden at 45. So that brings you to the conclusion that something else, apart from their DNA, is leading to different outcomes. And that was actually done with rat experiments. They got the exact same rats with the exact, you can control the environment with the exact same DNA because they're inbred and with the exact same exposures, yet they both developed separately. So there's something else leading to this. And what we also know is, unlike people sitting here, one can differentiate based on genomics or Y chromosome, X chromosomes, those who are born male and those who are born female. Yet we also know that reptiles, if you take their DNA, you're unable to differentiate. Yeah, so it's a quite interesting thing. The reason being is the differentiation is not based on DNA. For a crocodile to be a male or female, it's based on the exposure of the temperature, the egg's temperature, at once the egg is laid. So an increased temperature would lead to men, a reduced or males, and a reduced temperature would lead to females. So we know that this has a direct effect, the environment. Here we're only talking about temperature. Yet over the last, I'd say 300 to 400 years, evolution was going at a particular pace. So humans were, well, technically if you're an evolution believer, you'd say, over 4 million years, our genes were based on whether it's mutations or whether it's polymorphisms or whether it's over a standard pace. Yet the human evolution, in terms of technology, since mechanization, the way we live, congregations, bigger cities, ACs, change of air, medications at much higher doses than we're used to, different foods that we're eating, sugars that we never had, prosperity. We're living longer, for certain. Just until the 1970s, I think our lifespan was expected in this region was in the 40s and 50s. Although we're living longer, we cannot argue that we're exposing and being exposed to new environments, which we understand that this will affect us, but we don't understand exactly how. And to appreciate that, we need to ask why and how. And that's the whole purpose of this whole business unit that we're working on in M42.
That's exactly where I was going to lead to. I think we know that it is causing problems. It is changing our genomics and epigenomics. But the question is, what is triggering all of this? What exposure is this leading to? So I think how we can correlate all the data sets we have, and this is the unique thing about being in the M42 umbrella, is that we have these data sets independent of each other. And if we can bring them together and somehow work out why this change in our genomic is happening due to this environmental cause, I think this will be a very powerful tool that can be obviously scaled in many areas. But Dr. Andrew, maybe if I ask you on, do you see over sort of your career that spanned in occupational health, changes that has supported improving the epigenetics? Have you seen any examples of this?
Yeah, absolutely. So we know that benzene is actually very powerful at demethylating the epigenome. But the oil and gas industry has been amazing at moving to closed systems and risk managing that. So we used to have higher rates of leukemia in employees who worked with benzene. But now we don't see that. They're actually, you get this healthy worker effect. They're actually sometimes healthier than the general population, even though they're being exposed to a very dangerous chemical. And this is what we've learnt through occupational medicine, is that you don't have to just accept your fate. You can use masks, you can use air filters to make clean air. You can use filters to make sure your water's clean, to get the PFAS and the plastics and the pesticides. You know, you can choose healthy food. You can actually choose to be healthy. And that's what occupational medicine has taught me. So I've seen these very unhealthy workers actually, through good occupational health and safety practices, become really healthy workers and be contributing to society, keeping their families well, teaching them to be good citizens. This is why education is so important. If you really want to learn about the epigenome, I urge you all to read about carcinogens, or all to read about risk management, and use that on yourself. Actually use your knowledge to improve yourself, your family, and then go and improve society with that knowledge.
Absolutely. I think what people represent or misunderstand is that you may think that there's a disease-causing agent here. So, benzene, asbestos back in the day, and what have you. But the reality is that that agent is causing a change in your genomics that's causing the disease, right? So that's where we get into the correlation between an environmental stimuli and the effect on causing of diseases. But it could be changing your genomics in other ways that may not be a causative agent to disease today, but maybe a causative agent for many other things. So it's directly related to changes in our genome. It's very important to understand how even simple things like food affect our epigenetics as well. So Dr. Javier, if you don't mind, we need to make it simple. I don't want us to go into the science of all of this, please. For just, it's the end of the day, and we need to stay fresh and alert. But is there a way we can build an experiment and find a way to connect the two dots together? What actually in the environment is potentially causing these significant diseases within our communities?
Yes, we can, but one of the challenges of methylation is that it forces you to really understand what you want to study or to tackle. Because genomics is an easy shot, so to speak, because as I mentioned, genomics will not change over your life, and it will be the same if I take blood from your food or from other parts of your body. But genomics is not the case. So if you want to study how, certain alcohol or being a firefighter affects the epigenetics of lungs, because presumably that's what will end into disease, or liver, then you need to design that experiment to assess the methylation in those organs, because maybe how methylation patterns are in your food are relevant. Just to give you an example, so if I sequence your genome, probably our ancestry will differentiate us, but when you go into the epigenomics, probably all epigenetic marks from our liver between you and I will be more similar than if we were to sequence or the epigenetics or stomach versus another stomach. So that's yes, to answer your question, we can do a design experiment, but we really need to, there's no one experiment to carry out, but one that we need to think carefully.
As always, you made it very much more complicated than it has to be, but thank you, Dr. Javier. No, I'm just joking. Dr. Wael, let's see how we can use AI now as a tool. We're sitting on all this data that comes from the environment, both from a wastewater analysis that's obviously been excreted from our bodies into the sewage system, all of the testing that we do at our central testing laboratory of all the products that are imported and manufactured and digested here in our communities, and as well as all the genomic capabilities that we have. Can we find a way to build an AI model or allow us to, first of all, interpret this significant amount of data, large data scales, but correlate them and find insights that could hopefully expedite such experiments and allow us more insights?
So, full disclosure, AI until recently for me was artificial insemination. Yeah, so that's the first disclosure. Yeah, I've got a lot of very smart people sitting around me though, who help with designing experiments. So we have a full team, and we have a lot of discussions about how can we utilize and leverage the available machine learning. But if you take it back to simple statistics, what we have is a lot of data points now. We have a lot of data points that could be scraped from a lot of different places. And by scraped, I mean, if you look at your own phones all the time, they're collecting the temperature of where we are today. You've got you've got watches, you've got sensors all around, getting oxygen quality monitors. We've got, and we also have a big data set of clinical records at the moment, in particular, whether it's in the UAE or outside. There's also a lot of eventualities and events pre and post. So can we utilize and leverage all of that to see retrospectively or prospectively how that would affect possibly? Do we have that? Are we there yet? I'd probably say we're a little bit further away from that. And the main reason being is we don't understand epigenetics to its fullest in itself yet. What Javier mentioned was methylation. And methylation is only one form of epi. And epi means more than genetics. So we know genetics, and by genetics we mean the genome and the sequences. Epigenetics is more than genomes. You've got the histones, you've got the way it's tied. And again, a very simple example is butterflies, you know, the way they stage from 1 to 1. They have the same genome, yet the larva, through the pups, until it becomes a butterfly, is still the same. And we don't yet know which exact variable is the one causing the effect. And I think what is important is for us to realize that life expectancy has improved over the last 3 or 4 decades. So we're living longer. So the question is, how can we leverage everything to live longer, better? And I think we are moving forward very quickly with support of whatever tools that we have available to us. AI being one of them, and very smart people who know how to function and operate AI being much more important, of which we have a plenty in M42.
Thank you, Dr. Weil. Now, every time I think of AI, I'm going to think of that comment you just mentioned. So thank you for that. But yeah, no, on a serious note, I think this is an ever-evolving discipline and science, and I think we're just literally scratching the surface of this. I think we've had really important discussions over the last 3 days on everything linking to health and healthcare, but obviously we coin it as health because we want people to live longer and healthier. So I think once we crack the code or understand how the environment is directly affecting our health, I think this will be a day where we can really start putting measures in place. We are doing so, but without understanding the true causing agents and how it's affecting us. So on that note, on that positive note, I'd just like to thank our panelists and the audience for their patience. And again, a round of applause for our panelists, please.
Any questions?
Thank you. Fascinating panel, Elliot from Chromodiversity. A very general question. What is today's best sense of the proportion of the genetic code impact on health versus the epigenetic expression in overall health? And then secondly, regarding mental health, how far advanced are we in understanding the epigenetics of the most complex organ in our bodies, the brain? Because perhaps the relationship between smoke and lungs is a simpler one than the relationship with schizophrenia or other mental illnesses.
I can answer that because I've got some stats right here. So they say 20% of cancers, 31% of cardiovascular disease, 42% of strokes, and 35% of lung disease, COPD, is attributed to environmental factors. So it's actually a really large amount of your diseases out there is caused by the environment. But I have this saying that says, every disaster is an opportunity. I had this brilliant occupational physician, and he worked with vinyl chloride, and they all got the workers got liver cancer. But he studied that vinyl chloride. He realized it turned on an oncogene, and then he realized that he could bend the protein and he could turn off that oncogene, and it led to a potential cure for one of these cancers. So every environmental carcinogen is actually an opportunity. I'm studying long COVID. The opposite of fatigue is energy. Who here would not like more energy? So when you think about all these environmental dangers, I wouldn't think of it as an opportunity. Every time you find an environmental danger, one, you can manage that risk so you can be healthier, and two, you can study that risk and work out how to reverse engineer it with artificial intelligence to actually find a cure. But on your second question on mental health, there's certainly a strong environmental link there. And I've been working with hyperbaric oxygen therapy in long COVID. And again, we're seeing that you get neurocognitive benefits in people with chronic traumatic encephalopathy. You can actually see cognitive benefits from hyperbaric oxygen. So again, every disaster is an opportunity. Now they're showing that through long COVID, that you get better cognitive effects, and they're using it on other people with mental health illnesses and finding that positive effects as well. So there's some really exciting research going on, but it is about working as a team to get all this knowledge and look at these opportunities, because they are. Every disaster is an opportunity.
Thank you, everyone, again. Thank you.
much. Let's ensure that we get a wonderful group photo before we release you from the stage. Thank you so much, Elbara, with our wonderful panelists. We're going to set up for the very last panel discussion, which is our penultimate session today. So while we do that, please give them all a big round of applause, and we're back with you in just 2 minutes' time. Thank you so much.